You might have watched Nickelodeon TV as a kid. Well, now welcome to Nickel NTA TV. So basically, what this in this column is filled with these little beads that are attached to nickel, and they're going to help me purify my protein. So I'm using it with this Acta purifying machine, but you can also do it gravity flow, where you have resin and then you put it in one of these tubes. But I'm going to use the Acta today. So basically, I was just showing you that column, but I'm using this column. It's actually the same type of thing, it's just attached in here. And these loops are going to help direct my protein solution into and through the column. And then later, when I want to elute it, it'll come out in this little part here. So when I'm able to use this column because I expressed my protein. So when I had the cells make my protein, I had them put on a his tag on the end. So it's just a tag of 8 histidines in a row. So histidine is just an amino acid, which is a protein letter. We'll talk about all this more later in the video. I just want to give you a look at what it looks like in practice. So I had these bacterial cells make my protein for me. And then I broke the cells open, which we call lysis, and then I spun them down in a centrifuge, and so to pellet out like the cell wall, so the cell membranes and all the other insoluble gunk stuff. And so then I took the liquid from that and I filtered it. And so I just like squished it through this syringe filter, because when I'm using this Acta, I don't want to clog up the tubes, because that would be bad. So I filtered it, and now you can see that, so I actually have 2 samples going because I'm testing out a couple of different, so this one was expressed in TB and this one was in LB, and so I'm trying to see if either of them worked. And so I have a post on those. Those are just different types of bacterial media, so bacteria food. So I tried growing cells in different types of food to see if one would work better. So basically, the machine is going to pull up, it's right now, what it's doing is it's pulling the sample up through a column, or through that tubing, and putting it into the column. And so this column is filled with these beads that have this linker on them that clamps down on the metal nickel. So you can do a similar thing with cobalt, as we'll talk about later. But basically, the nickel will then, so it's stuck, the nickel stuck to the beads, and then the his tag that you put on your protein, it likes to bind to nickel. And since you have all these hisses, what it can do is it really clamp down. So it's like it is a polydentate ligand, or polydentate chelator. And so a chelator is something that bites down or claws down on a metal, and so you have a bunch of his in a row, so it can bite down in many places. So it's polydentate. And so it's going to latch on to that, whereas if you just had like normal histidines hanging out around the protein, they're not going to grab on as tightly because they don't have that like bunch of histidines in a row to clamp on. And so that's how you're going to get the selectivity. But there's still going to be some stuff that's like non-specifically binds. So what you do is you have, so we're gonna push the protein off later. So the idea here is we want to get our protein stuck, wash everything else through, then get our protein unstuck by itself. So in order to get it unstuck, we're going to use a competitor called imidazole. And so imidazole is going to, it looks like histidine, so it's going to compete it off. And you can use like a higher concentration of it. So I have these 2 buffers. So I have a high imidazole and a no imidazole. And so these are the same basic buffer, but one has imidazole, a high concentration of imidazole, and one doesn't. And then the Acta has these pumps that it will mix the 2 of them. And I have it set on this system. So I have it set in this program where it's going to do this big long wash. So it had this equilibration step where it just basically got the sample, got the column all filled with a low level of imidazole buffer. So a buffer is just a salt stabilized pH, like salt stabilized water, pH stabilized salt water. And so I had the equilibration, and now it's putting the column on, the sample on, and then it's going to do a wash step. And so this is all happening at the same low concentration of imidazole, so a low concentration of the competitor to keep off really non-specific things, but you don't want to wash your protein off in the process. And then it's going to try these 3 different, so it has a couple different concentrations that it's going to try for the elution. And then as a last step, it just has like 100% of so 500 millimolar imidazole. So I'm expecting it to elute probably at this 250 millimolar imidazole. But you want to have this high imidazole wash at the end to wash off anything that's any proteins that are still stuck on there, so that the next person that goes to run the column isn't going to get a surprise and have a bunch of crap come out that was left on the column from the previous person. It's happened to me before, and I was like, what the heck is this coming from? And it was apparently that the person before me hadn't done that wash at the end. So do the wash. So right now I'm just in that wait and see mode, and I'm hoping that this is going to work out. And so now let's get into the details about what's actually going on. If you're watching on the Instagram version, this is going to be longer than I can fit here, so visit the YouTube or my blog to see the full video. One of the most common methods used to purify proteins is affinity chromatography. The idea with chromatography, as I've gone into more detail in past posts, is that you have a column filled with little beads called resin, and that resin has different properties, and those different properties will affect how they interact with different proteins based on those proteins' different properties. And so you flow this mix of proteins through this column, and the proteins interact differently with the resin, and so they will travel differently through the resin. Either they'll go slower or faster, or they'll get stuck or they won't get stuck. And so the idea with affinity chromatography is that you want your protein to get stuck. And then you'll let everything else flow through. You'll wash it and wash it and wash it, and then you'll let it get unstuck. And then it's just your protein alone. So you unstick it with a competitor. So you stick it, then you wash everything, and then you add a competitor that looks like it, and it pushes it off. So we talked in a previous video about strep tag, which was one form of affinity chromatography, and today I want to tell you more about the his tag, which is used for IMAC, Immobilized metal affinity chromatography. And so this is just an overview, is that your resin, so those little beads, often they're like NTA, which is just this chelator, so this metal binder. We'll get more into chelation later, but it's attached, it's grasping on to a metal, and so this is often nickel, or sometimes cobalt. Your protein of interest, you design it so that it has a histidine tag at the end, so like 6 to 8 histidines in a row. Don't worry if these words don't make sense yet, it'll make sense later hopefully. So you have this tag on your protein, and that's going to bind the nickel. You let everything flow through, and then you push it off with imidazole, which looks like the histidine, but it's just this like soluble part, and you can really flood it. So how do you get the his tag on the end? So when we're doing this, you can use, so if your protein has a lot of histidines, you can use that naturally, but it won't work as well. But if you have, when you're doing recombinant protein expression, you're basically sticking the genetic recipe for making a protein into cells like bacterial or insect cells, and then getting them to make the protein based on the instructions that you put in. And because you're putting in the instructions, you can add a little extra onto the end, and this allows you to add the histidines, because histidines are just an amino acid. And then you express the protein, you break those cells open, you spin them down to separate the gunk parts from the soluble protein, and then you can do your chromatography. And so the affinity tag purification is often the first step in a multi-step purification regimen, because you can really snatch out your protein specifically, but it's from all the other stuff. And so it's more specific than things like charge or size, which lots of proteins will have similar charges or sizes to the protein that you have. But if you're able to weed out most of them through the affinity capture step, then you can you'll have fewer contaminating proteins for the further steps. And so the chromatography, so that we're going to talk about affinity chromatography today, but there's also ion exchange chromatography where you separate based on charge, and size exclusion where you separate based on size. So going back to what the hist tag is. So as I mentioned, histidine is an amino acid, and an amino acid is a protein letter. And so proteins are basically long strings of amino acids linked together through these special bonds called peptide bonds. So you get this long chain of amino acids called a polypeptide, which folds up into this 3D functional state. And so the way it folds up is based on like the different properties of the amino acids. So there are 20 common genetically encoded amino acids, and they have different properties. Based because of their unique side chains. So if you think of a charm bracelet, they all have this generic chain part, but then a unique charm hanging off, and that's like your side group. And so some of these are small, flexible, some of them are big and bulky, some are positively charged, some are negatively charged, and so these groups have different preferences for where they want to be in the protein, like who they want to hang out with, do they want to be near water or not, and so this influences how the protein folds up. But, and so histidine is one of those amino acids. And so we can, if we stick the genetic instructions for making histidine, if we stick like 6 or 8 of those in a row in front of the genetic recipe for our protein, it'll get added onto the end, and then we can do our affinity chromatography. So what's so special about histidine? So this is showing the R group of histidine, highlighted in blue. And you can see it has this weird like ring structure, and there's that weird like dotted lines. Or you can also show it as, there are different ways to represent this, but what it's showing is that you have resonance stabilization or electron delocalization, or since this is in a ring, aromaticity. And so I'm going to tell you more about that later, but basically it means that there's going to be this kind of like donut-like ring of negative charge above and below the histidine, which makes it really attractive for the positively charged metal. And so then we can compete it off with imidazole. And then we often design the hist tag so that there's a protease recognition site. So a site that a pair of protein scissors recognizes and cuts at. So that way, after you get your protein off, you can then cleave that tag off so you don't have to worry about it anymore. And then you can focus on separating by charge and size, which don't require you having that tag there. So as I mentioned, there's that weird like structure thing going on with histidine. It has to do with how the electrons are shared. So atoms, like individual carbons, hydrogens, nitrogens, they're made up of smaller parts called subatomic particles. There's protons, neutrons, and electrons. And so the protons and neutrons are held in the central core, and then you have the sea of electrons hanging out around it. And these electrons are negatively charged, and the protons are positively charged, and the neutrons are neutral. So if you have an imbalance of protons and electrons, then you get a charge. And so this is how we get like metal cations, so positively charged molecules. They have more protons and electrons, so they're positively charged. So atoms form bonds to form molecules by sharing pairs of electrons. And so typically one molecule will share one electron, and the other one atom will share one electron, the other atom will share the other, and you get this pair, and they call that a covalent bond. And if they share in a double bond, you have 2 pairs of shared electrons. Triple bond, you have 6. And electrons can also exist as unbonded lone pairs, like we saw on the ends. So the nitrogens often have these lone pairs of electrons. So something called resonance stabilization happens when multiple atoms are sharing electrons. And so it's not just 2 atoms involved, it's multiple atoms involved. So it's kind of like this electronic commune. And when this happens, it's like the super stable situation that molecules really love. And then if it happens in a ring, so most classically in the benzene ring, you get this thing called aromaticity. And so what's really cool about this is that those delocalized electrons, so those like communal electrons that are shared, so those extra ones, they actually kind of like form these donuts above and below the ring. So you have these like negatively charged donuts, which you'll really like to bite onto the metal. And what's even cooler is that with histidine, you have those nitrogens, which have the lone pairs. Lone pairs can do something called form a coordinate covalent bond. So in a coordinate covalent bond, instead of having one atom share an electron, each atom share an electron, you have one atom share 2 electrons. And so you need something with a lone pair, and so nitrogen can do that. And it can form these coordinate covalent bonds with the metal. And so when we have something bind to metal in multiple places, we call it a chelator. So chelate is like claw or bite. And so going back to that bite, so the number of bite marks basically is how dentate it is. So if you have a bidentate thing, that will be able to bite down 2 places. Tridentate, you can bite down 3 places, etc. So you might have heard of chelators like EDTA that you use in the lab sometimes to kind of hide metals from things like proteases, non-specific proteases that we don't want chewing up our protein, or that sort of thing, or RNases or DNases. And so when you have a bunch of histidines in a row, they can serve as a polydentate chelator. So they have multiple bite sites that they can bite down on the metal. So the metal has these like big electron clouds, so they can get bitten down on in multiple places. So when you have a his tag, it's like you have this polydentate chelator attached to your protein. And then that metal is actually held to the resin with a chelator that's attached to the resin. So the resin basically has this chelator, and the metal is just attached to the resin. And so that's a stronger bond than the his tag in your protein and your resin. So basically, the metal is held by these by chelation, so by these coordinate covalent bonds onto the column. And so that's not a permanent interaction, but it's stronger than the interaction between your protein and the resin, and so and the protein and the metal, and so that allows you to wash off your protein without having to worry about washing off the metal. But you can also wash off the metal, or add more metal if some of it is like leaching off. You can add more to recharge the resin, so use like a nickel chloride solution. It's pretty cool because it starts off looking all like green, but then as it goes through the column, then it gets like bluer and stuff. It's pretty cool. But anyways, you can also change the metal. And so I was most commonly use nickel, and so that's your blue. But you can also use cobalt. And so this is when I was trying cobalt a couple years ago, because I had a protein that wasn't working well with the nickel. And so why might cobalt be better? So cobalt is so it's a similar like cation to the nickel, so it can bind to the his tag, but it's pickier. And so it's less likely to bind non-specifically to proteins that like just happen to have a bunch of histidines or something. But it also has a weaker affinity, but doesn't bind as strongly. But so sometimes cobalt resin can be good to try. So like I think that it's called like commercially they sell it as like talon resin. But yeah, so you can use this. I just use the same NTA agarose NTA resin, and so NTA is that chelator attached to the beads themselves are like made out of agarose, and they have this NTA linker. There are also some other linkers and that sort of thing, which I'm not going to get too into here. I just wanted to introduce that to you. And yeah, so this is similar in concept to when we were talking about affinity, chromatography in terms of the strep tag. With the strep tag, instead of having the same amino acid, like a string of the same amino acid, we had this string of different amino acids that had this specific sequence that make this, that bind specifically to this streptavidin, a streptavidin mimic resin. And so I have a post on that if you are interested in learning more about that as well. And so that is IMAC, and I hope I've convinced you that it's even cooler than the latest Apple product. Hope that helps.